Okay, hi everyone. Welcome to this webinar. On behalf of the Biochemical Society and Portland Press, I'm very pleased to welcome you all here. And this webinar is part of the Biochemistry Focus webinar series, which includes different research topics of different research areas in molecular biosciences and practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please have a look at the BiochemSoc website for more details on that. And you're free to submit questions throughout the session, and they'll all be dealt with at the end. Hopefully we'll have plenty of time for some good discussion. So I'm Naomi Pollock. I'm actually have just finished as a postdoc at the University of Birmingham, and I'm about to start a new job in a couple of weeks' time, starting my own research group. So I've also been on maternity leave for the last year. So this is a really great opportunity for me to immerse myself back in the wonderful world of membranes. So I've already said questions will be asked at the end. So I think I just need to introduce our first speaker and then hand the floor over to her. So the first speaker this afternoon is Larissa Socrier from the Max Planck Institute for Dynamics and Self-Organization. Larissa studied in France, where she obtained her Bachelor of Science degree in the field of biology at the University of the French West Indies. She got her master's degree in biochemistry at University Planck, and a PhD in biotechnology and biophysics at the University of Technology of Compiègne. She's currently a postdoc at the Max Planck Institute of Dynamics and Self-Organization in Germany. Her research focuses on membrane biophysics, and she uses GB3 glycolipids to investigate membrane domains and proteins, and glycolipid interactions to comprehend the mechanisms leading to the internalization of toxins into cells. So I'm really looking forward to hearing your talk, Larissa, and it's over to you to start now.
Thank you for the introduction. Can you all see my screen and hear me properly?
I can, yeah.
Okay, so as it was said, in my research project, I investigate glycolipid bacterial proteins interactions. And today I'm going to explain to you how I investigated the impact of GB3 structure on membrane organization and Shiga toxin B interaction. But before getting into that, I would like to give you a few reminders about biological membranes. As you know, they are very important because they are involved in several physiological processes. For example, they constitute a barrier that allows to separate the inside of the cell from the outside of the cells. They are also involved in compartmentalization and signaling pathways. And you can see here a general representation of the structure of the membrane as it was depicted 50 years ago. You can notice that lipid and protein are evenly distributed in the plane of the membrane. But today it is widely admitted that membrane present a high lateral heterogeneity due to the ability of lipid and protein to self-assemble into domains that are often called raft. And because biological membranes are so complex, various artificial systems have been developed in order to study membrane properties, as well as lipid-lipid and lipid-protein interactions. You could see a few examples here: Langmuir monolayers, lipid vesicles, supported lipid bilayers, or even pore-spanning membranes. These are the ones that I mainly use in my research topic. And it was found that certain lipid mixtures containing cholesterol, DOPC, and sphingomyelin have the ability to phase separate into ordered domain in dark and disordered domain in gray. And the idea that the ordered domain would replicate the properties of lipid raft has gained in popularity in the biomembrane community. So in order to study domains in artificial and cell membranes, various modified lipids have been developed. You can see here these free lipids that partition either in the LD phase or the LO phase, depending on the structure of the fluorophore. We can also cite membrane polarity sensitive probes, which we can use to calculate the generalized polarization parameter. This is a parameter that reflects membrane order. In our lab, we have focused on GB3 glycolipids, but recently photolipids have gained popularity as nanotools in order to study domains and membrane properties. And the particularity of these lipids is that they possess a light-sensitive azobenzene moiety that can change configuration from trans to cis upon light irradiation. And the change of configuration, which is also called isomerization, will modify the global geometry of the photolipid from a cylindrical shape to a conical shape upon UV irradiation. And this is something that is reversible upon blue light irradiation. Photolipids have been used to study membrane properties, in particular its lateral organization. And you can see here this example study that was performed in the group of Professor Dr. Trauner. Basically, they inserted photophosphatidylcholine into phase-separated giant unilamellar vesicles. And you can see that upon UV irradiation, so isomerization from trans to cis, you could see a demixing, so a disappearance of the LO domains. And that was something that was reversible upon blue light irradiation, so isomerization from cis to trans. And they were able to reproduce these experiments several times. A similar observation was done with another type of model membrane, here supported lipid bilayers. And you can see that the area of the LO domain in gold diminishes upon UV irradiation, and that is something that is reversible upon blue light irradiation. You can see that the area of the LO phase is becoming bigger, and you can notice the appearance of several gold little dots in the LD phase of the membrane. However, these studies were conducted in pure lipid systems, and given that eukaryotic plasma membrane contain approximately 60% of protein by weight, this raises the question of what would happen in presence of a protein. Is it possible to rearrange membrane domains like this in presence of a protein, and what would be the impact on the protein density on the surface of the membrane? So, in order to address these questions, in collaboration with the group of Professor Dr. Daniel Wirtz, we have designed a new set of photo-GB3 glycoconjugate, which constitute a natural binding site for the bacterial protein shigatoxin. And basically, on the fatty acid of GB3, we have attached the azobenzene at 4 different positions, which should allow us to modify more or less the geometry of the photolipid upon light irradiation. So the first thing that I did with these compounds was to investigate their impact on domain organization in absence of protein. So to do so, I prepared phase-separated supported lipid bilayers in which I introduced an important amount of photo-GB3. Then I irradiated the membrane with UV, then blue light, and then I observed the membrane by fluorescence microscopy, and this is what I saw. You can see that upon UV irradiation, the LD phase marker in gray slowly diffuses into the LO domain in dark. And upon blue light irradiation, you can notice the formation of those black little dots in the LD phase of the membrane. And you can see here detailed images of the experiments. You can see that approximately 8 minutes after UV irradiation, the LD phase marker has diffused into the LO domains to form these structures to which we refer to as LD lakes. And immediately after blue light irradiation, you can notice the formation of those black little dots that we term LO lakes. So in order to better comprehend the impact of these compounds on domain organization, I first focused on the cis isomerization cycle, so irradiation with UV. And then I calculated the area of the LO phase that I divided by the area of the LD phase, and I reported the ratio as a function of time. You can see here detailed images of the previous experiments, and you can see that following UV irradiation, the ratio at first increases, which would suggest in an increase in membrane order. This is because the LD phase marker slowly diffuses into the LO domains in dark. And then when the LD lakes become discernible, the ratio diminishes, indicating a fluidification of the membrane upon UV irradiation and thus trans to cis isomerization. And I did the same analysis with all the photo-GB3, and you can immediately notice that the deeper the adsobenzene, the stronger the fluidification of the membrane is upon UV irradiation. So following this analysis, I then focused on the blue cycle, which turned out to be more challenging to characterize. And to do so, I prepared a supported lipid bilayer as previously described, and I fluidified it by irradiating with UV light. And then I had to perform multiple blue light irradiation cycles, and this is what I saw. You could see that irradiation with blue light triggers the formation of several LO lakes in the LD phase of the membrane. And this is something that I was able to reproduce several times following blue light irradiation. And here you can see detailed images of the experiments with the presence of the LO lakes in the white rectangles. And I calculated the LO-LD ratio at each time point of the experiments, and you can notice an increase, which indicates an ordering effect on the membrane. And then here, to better characterize the reaction and its impact on domain organization, I superposed the images at T0, so before blue light irradiation, and at T40 minutes after the multiple blue light irradiation cycle. And you can notice in green on the superposition the increase of the area of the LO domain, indicating an ordering effect on the membrane. And to compare all the four photo-GB3, again, I calculated the LO phase area that I divided by the LD phase area. And this time I calculated the variation, so the increase of the ratio after 40 minutes. And you can see that the deeper the adsobenzene, the more pronounced the increase is, so the more pronounced the ordering effect is following blue light irradiation cycle. So following these experiments, I focused on lipid-protein interactions, and to do so, I prepared phase-separated supported lipid bilayers as described previously, and then I incubated the bilayers with shigatoxin B. And as you can see on this image, the protein behaved naturally in presence of the photo-GB3 as it was bound to the LO phase as described elsewhere in the literature. I then performed the UV and then blue light irradiation cycles and observed the bilayer by fluorescence microscopy. And this is what I saw. You can see that following UV irradiation, there is the formation of granular structure in the LO phase of the membrane, and that this structure persists in the LO domains following blue light irradiation. And here you can see detailed images of the experiments and the presence of these grainy structures in the LO domains despite the blue light irradiation. And to get a better view on these structures, I then zoomed on a portion of the membrane and I recorded a Z-stack, and you can clearly notice the formation of these structures that we called protein lakes. Following these experiments, I exported the experimental setup on the AFM microscope with the objective of getting a better resolution on the protein lakes. So here you can see an image of a phase-separated supported lipid bilayer that are incubated with shigatoxin B. And you can see the height profile analysis on the right side. You can notice that the height difference between the LO and the LD phase is very large, is approximately around 3.4 nanometer, indicating a successful protein binding. In absence of protein, the height difference would have been around 1 to 1.5 nanometers. Following that, I performed the UV and then blue light irradiation cycles and recorded several images over time. And this is what I saw. You can see that initially the surface of the domain is homogeneous, but approximately 10 minutes after UV irradiation, you can notice the formation of the protein lakes and that they persist despite blue light irradiation. You can see the detailed images of the experiments, and you can notice the appearance here of the protein lakes 10 minutes after UV irradiation, and that those structures remain on the membrane despite blue light irradiation. And to get a better view on these structures, I lowered the speed of acquisition and I zoomed on another domain successively and recorded these images here. And you could see that I was able to see that they have a size comprised between 10 nanometers for the smallest up to 500 nanometers for the biggest. So to conclude, in absence of protein, when it comes to the impact of the compounds on domain organization, concerning the trans-to-cis isomerization, so irradiation with UV light, I was able to show that it triggers a demixing, so a disappearance, a diminution of the area of the LO domains, and that this effect is more pronounced when the adsobenzene is deeply embedded into the membrane. Conversely, when it comes to the cis-to-trans isomerization, I was able to notice an increase of the area of the LO phase, indicating an ordering effect on the membrane. And again, this effect was more pronounced when the adsobenzene was deeply embedded into the membrane. Concerning lipid-protein interactions, the presence of the adsobenzene on the fatty acid of GB3 did not alter the behavior of shigatoxin B, as the protein was bound to the LO phase of the membrane following incubation with the samples as described in the literature. And isomerization from trans to cis led to the formation of protein clusters which persisted on the membrane despite blue light irradiation. So to finish, I would like to thank my advisor, Professor Dr. Claudia Steining, who gave me the opportunity to conduct this research project. I also want to thank the students and the technician that assisted me with the experiments. I also want to thank our collaborators for providing with the GB3 derivatives, the protein samples, and for doing additional biophysical characterization. I also want to thank the funding bodies for financial support and these institutions here that provided me with travel grants, which allowed me to present my work in several conferences. And finally, I thank you for your attention.
Hi, I am back, sorry, I couldn't see myself. Thank you so much, Larissa, that was really fascinating. I have many questions which I will save up for the end, as long as everyone else's questions are answered as well. Thank you for sharing that with us. So our next speaker today is Adam Wells from the University of Oxford. He completed his BSc in Biological Sciences at Durham University, and he's currently a PhD student in Professor Clive Wilson's lab, studying the molecular regulation of exosomes, which is a class of extracellular vesicle that functions in cancer signaling, neurodegeneration, and many other pathways. He is due to complete his PhD very shortly in the coming months, and he hopes to continue as a researcher either in this field or in EV-based research technology. So, Adam, I can hear a little bit of feedback, but I think I'll be muted in a second, so hopefully that will work nicely. Thank you for being here, and I will hand over to you.
Thank you, and let me know if there's any feedback or other issues. Hopefully you can see my slides. Yes. Okay, I'll assume that the slide.
Yes, I can see them. Sorry, I was taken offline, but yeah, they're all good.
Great. Well, thank you very much for having me to talk today. I'm going to be talking about, as you said, my research into the formation of secretory granules and exosomes within intracellular compartments. Unfortunately, I can't see my little prompts along the side, so if I have a pause, that's just because my brain is slow. To begin with, intracellular compartments. I maybe don't need to say this, but intracellular trafficking is a fundamental part of eukaryotic biology. It determines what cells can uptake, secrete, degrade, and by extension, therefore, it plays a major role in cell-to-cell signaling. And indeed, eukaryotes have developed and evolved quite a range of intracellular trafficking pathways, some of which are shown in a fairly busy slide, sorry, in a fairly busy diagram just here. Now, although there are many of these different pathways, they share, for our purposes, one key characteristic, which is that they are all comprised of small, nanoscale vesicles for the most part. And that's really interesting, but it's also very difficult for the field because they're simply difficult to study. Imaging such small vesicles is especially difficult without use of electron microscopy, and chemically isolating just one particular population of vesicles is also obviously very difficult. And that leaves us in a state where, despite many of these things being very important, we actually still don't know a lot about the fundamental biology within cells for some of these. And as such, my research focused on two important but not that well understood structures, which is secretory granules, formed as part of the regulated secretory pathway, and exosomes, which are formed as part of the endosomal pathway. So again, maybe this doesn't need to be said, but secretory granules and exosomes, they are important for signaling. Secretory granules are formed as part of the regulated secretory pathway that transports materials from the Golgi directly to the plasma membrane. And as part of that, proteins, hormones, signaling factors such as that, they aggregate into insoluble granules that you can maybe see in this pathway. Oh yes, and famous examples of this pathway are going to include ones that you'd know: insulin, adrenaline, neurotransmitters within the brain, specifically neuropeptides, and many others. And in fact, in recent years, dysfunction in secretory granule formation has been implicated in diseases such as diabetes and neurodegeneration. Exosomes, meanwhile, quite a different structure. They are a form of extracellular vesicles that get formed within the endosomal system. So this is actually an offshoot of the endolysosomal pathway, but these structures are 30 to just 150 nanometers. They're therefore particularly difficult to image with anything other than electron microscopy. But what's interesting, and the reason that they have received so much attention in recent years, is that during their formation, during which they are essentially, exosomes are formed via the inward budding of endosomal membranes, they become enriched in specific signaling factors, RNAs in particular, and proteins, and they can then effectively be secreted from cells and transplant those materials into other cells that uptake these exosomes. And that makes them potent signaling factors that are particularly prevalent in cancer biology, immune cells, and several other places, such as neurodegeneration. But both of these structures are generally very difficult to study, as I've mentioned, and that is where our research in the Wilson group comes in. Essentially, around 6 to 7 years ago, they sought to develop a system in which you could study intracellular compartments more easily, more readily, and more flexibly. And to do that, they set up a system within Drosophila. In particular, they used Drosophila secondary cells, which are these green secretory cells that are part of the male reproductive tract. And if you look at this zoom view, you can see why they're such an attractive model, which is that this is not an electron microscopy image, but we can see these compartments, and marked in green in a live cell, are secretory granules. These compartments are many times the size you'd find in other systems. And in case you were wondering, yes, these compartments also produce exosomes. As you can see, they're these internal puncta of light. In particular, they produce a special kind of exosome called Rab11 exosomes. They were actually first discovered in this system, but they've since been identified as promoting cancer signaling in human cancer cells. So alongside these the great advantage of these large compartments, we also have a few other advantages just by virtue of working in Drosophila, which is that we have cutting-edge genetic tools. We can manipulate individual cells, individual genes at specific times. And also, because they're so large, we can actually avoid using electron microscopy to image these compartments, and we can use fluorescence microscopy, which in particular allows us to use live cell imaging. And one of the developments that I spent my PhD trying to establish, time-lapse imaging. And in addition, it's an in vivo system, which is always nice, or ex vivo technically when we're doing our imaging. And so yes, we have these two structures. They are quite different, but that's how I'll be dividing my talk today. First of all, examining the regulation of secretory granule formation, and then Rab11 exosome biogenesis within this system. So the fun part, the results. Since this was a new system, one of the first things that we were required to do was characterize this system. And then a great place to start with that was studying Rab GTPases. Now this is a GTPase family of trafficking regulators related to, it's a small RAS-like proteins, and they insert themselves into lipid bilayers and regulate the fusion, transport, maturation of different intracellular compartments. And after studying many Rab gene traps, we noted that two in particular, Rab6 and Rab11, marked these large secretory compartments. And actually, they overlapped on many compartments, which led us to wonder what the difference between these two markers was. And it came with a little bit of closer examination. As you can see in this far left image, Rab6 is present on several compartments with secretory granules in them, but it also strongly labels this central compartment that has no secretory granule in it. Rab11, by contrast, exclusively marks secretory granule compartments, and actually, if you look at this graph, you'll see absolutely all of the secretory granule compartments are also labeled by Rab11. And this image shows that nicely. Although we have lots of overlapping compartments, where Rab11 is present in these yellow tinged compartments, you always have a secretory granule. And if you look at the 4 or so compartments marked by Rab6, they're all empty, which led us to wonder what the relationship was precisely between these Rab6 and Rab11 labeled compartments. And to do this, we carried out time-lapse imaging, which is something that simply wouldn't be possible in most other systems where you'd rely on electron microscopy. And so, perhaps this one won't, there we are. They started in the wrong order. But if you keep your eye, what we found was that the Rab6 labeled compartments, such as this one in the center here, directly transition into a Rab11 identity across a period of sometimes 8 or more hours. But in addition to that, if you view this compartment from the start here, you see that during this transition, the secretory granule is actually formed inside of these compartments. And in addition, for what it's worth, exosome biogenesis also seems to perfectly coincide with the start of this transition. And this led us to wonder just what the role of each of Rab6 and Rab11 and this transition was during compartment maturation. And so to investigate that, we took each of Rab6 and Rab11 and we knocked them down, specifically within secretory cells, within secondary cells, using a secondary cell specific driver to express an RNAi for each gene. And we did it twice. We did it firstly in a background where we had a fluorescent RAB11 gene trap, and again, then in a background with a fluorescent RAB6 gene trap, labeling their respective compartments. And these images are going to stay, this image panel will stay consistent across the next few slides, but the graphs will change to highlight the different things that we're looking at. So, first of all, when we knock down either RAB6 or RAB11, we see a sharp, very significant reduction in the number of RAB11 backgrounds. Of RAB11 compartments. And you can see that really just by looking at these images. These cells have particularly interrupted structures, and there's many fewer of these RAB11 compartments. That makes sense because RAB11 we found was downstream of RAB6, so it makes sense that both gene traps, that both genes are required for that RAB6, for RAB11 compartments to form via the RAB6 to RAB11 transition. And in addition, we find that the number of secretory granule compartments is massively reduced in following all knockdowns. And again, since RAB11 was a marker, we thought, of these secretory granule compartments, that fits. However, what was interesting was that when we knocked down both gene traps in the RAB6 background, we actually didn't always see a reduction in the RAB6 compartments. When we knocked down RAB6, of course, we see fewer RAB6 compartments. But comparing the control to the RAB11 knockdowns, we see that it actually did not disrupt RAB6 compartment formation. It could even increase it fairly significantly. However, this is interesting because even though we're still getting these regularly formed RAB6 compartments, if you recall, we still lose those secretory granules. And what this indicated to us was that this novel RAB6 to RAB11 transition that we had identified was necessary for secretory granule biogenesis. And in the absence of specifically RAB11 being recruited to these membranes, we got many of these compartments which were RAB6 positive, but which lacked secretory granules. And likewise, when we knock down RAB11, we also see that a lot of these compartments are still empty and they fail to form RAB6 labeled exosomes as well. And this leads fairly nicely onto the second part of the results, which is the formation of exosomes within these same compartments. So we found that RAB11 was required for these exosomes to form, but then I began to investigate the role of microdomains and lipid raft structures in exosome biogenesis. And this work really began when I was examining the distribution of a molecule called E-cadherin. Now that's a cell adhesion molecule. It has particular roles in cancer metastasis, and it's expressed in epithelial cells such as secondary cells. And so when I expressed a fluorescent RFP tagged form of E-cadherin, we found, as you can see in these zoomed images, that it co-localized with the membrane of these RAB11 compartments, but specifically it formed these small microdomain structures. And what was doubly interesting was that these microdomains were also co-localized with this, which is, as you can hopefully see in each of the zoom images, a developing, budding RAB11 exosome forming seemingly directly from this microdomain. Now this was particularly interesting to me. We ended up terming these microdomains ECAD lipid microdomains, potentially a temporary title. But we then began to investigate just what other factors could be clustering within these newly identified microdomains. And one of the first proteins examined was RAB19. It's another RAB GTPase. It's very poorly studied, but it's uniquely highly expressed in secondary cells. And yes, when we expressed a fluorescent form of YFP RAB19, we find that it does co-localize into these microdomains. And not just that, it can again be seen to be forming what appears to be the origins of a Rab labeled exosome, which generally we call Rab11, though in this case, obviously, it's being labeled by Rab19. And just to check this, again, what we did is we expressed YFP Rab19 with CFP Rab6, and we found that these chains, which can be identified in the Rab6 background, these chains of exosome precursors can be seen budding directly from microdomains labeled by Rab19. And so finally, we tested perhaps the most recognizable regulator of exosome biogenesis, the escort complex. Specifically, we examined a fluorescent form of the escort 0 protein HRS-GFP, and you'll be familiar with these results now. We co-express the two. We co-express HRS-GFP alongside a fluorescent ECAD molecule, and we find that they localize very nicely within microdomains just at the boundary of these compartments. And again, we see that these Rab6 labeled chains of exosome precursors are marked at their base by the protein HRS. And so as one of our to test the function of these microdomains and the proteins within them, we knocked down each of the three identified constituents of these microdomains, and we found that when we knock them down, you can see either by the graph or by the number of arrows which each label the exosomes within these cells. Yes, when we knock down any of these three factors, we lose the ability to form Rab11 exosomes, generally termed ILVs whilst they're within cells. And yes, ILV biogenesis is significantly reduced in the absence of these microdomains. And so there'll be just one more example, which is we became very interested in the role of signaling pathways in regulating exosome biogenesis. This is one of the big unknowns in the field. Although we know that important signaling pathways do control exosome formation, we really don't have mechanisms of how they're doing that. And so what we did is we examined the PI3K ACT-mTOR signaling pathway, an important cell survival, cell growth, cancer signaling pathway that had been identified as regulating Rab11 exosomes within human cancer cells. And we investigated in two ways. Firstly, we either decreased PI3K signaling, or we used constructs to increase PI3K signaling activity. And as you can see, when we reduce PI3K signaling, we significantly reduce Rab11 exosome biogenesis as well. Whereas, looking at these arrows, when we increase PI3K signaling in this pathway, we actually significantly upregulate the number of exosomes being produced. Now, that was fascinating by itself to me, but what was also fascinating was the fact that we could recapitulate that in terms of the formation of these microdomains. So again, if we decrease PI3K signaling, as you can see, even just by looking at the ECAD RFP channel here with these microdomains, you see that there's many fewer compared to the control. But if we upregulate signaling in this pathway, we actually artificially increase the formation of these microdomains, which led us to these conclusions. For the project overall, I think it's important. It was very interesting to find that a novel, non-previously identified Rab6 to Rab11 transition played such a key part in secretory granule formation and exosome biogenesis. But in addition to that, the formation of these microdomains appeared from our results to be directly tied to the formation of the Rab11 exosomes that we see budding from them in live cells. And the only real, and sorry, I should emphasize that whilst I went through the PI3K signaling pathway as an example, we have actually found other examples and other regulators that seem to match this pattern, where if we eliminate them and eliminate the ability of microdomains to form, we cease to form these exosomes. And so I think this offers a really interesting insight into a possible mechanism for signaling pathways controlling exosome biogenesis. And since this pathway, PI3K signaling, was identified in human cancer cells, there are, within the realm of exosome research, some really interesting parallels to cancer research as well. And with that, I'd just like to thank you for listening. I'd like to thank my contributors, and I'd like to thank the Biochemical Society for inviting me today and for supporting my PhD research. Thank you very much.
Okay, thank you so much, Adam. That was really fascinating, completely different from what I do, so I'm having to work very hard to take it all on board. But it's really cool actually to see two talks that are describing these kind of novel tools for investigating like really kind of fundamental things in biochemistry. That's kind of similar to the type of work I do, although in a very different area. So it's really nice to feel like there's just such a diversity of this type of work going on. And also thank you so much to both of you for beautiful talks and like very beautifully delivered on time. So we've got plenty of time for discussion if the attendees want to submit their questions. We have a couple already for Larissa's talk. I think people are probably still thinking through what Adam's told us about. Hopefully we will get some more questions in the next few minutes. So I think I might start by reading out one of the questions that someone in the audience has submitted for Larissa, if that's okay. So the question is, oh, in fact, no, there was someone who submitted two questions, I think. Sorry, I have two different tabs with them on, so I'm just finding the right one. Okay, so this says, great talk. I have two questions. The first is, can this be applied to in vivo models and act as a traceable tool?
I suppose the viewer is talking about inserting photolipids into biological membranes, right?
That's my assumption. Yeah.
To my knowledge, that would be something that would be difficult, because as you can see, to trigger the trans-to-cis isomerization, we need to use UV light, which has deleterious effect on cells and can cause cancers. But something that is currently being developed is red-shifted photolipids, which can be triggered by light irradiation with red light. That way we can shift the wavelength that would be required to trigger UV and to trigger, sorry, trans to cis isomerization and then cis to trans isomerization. So application in vivo appears difficult because of this drawback with the UV cycle.
Okay, thank you. And the second question is there strong evidence that the fatty acid alters the binding behavior of Shiga toxin subunit B, as well as the intracellular routing of the Shiga toxin GB3 complex? And if so, do you know any examples of that?
Yes, in our labs, we have synthesized various GB3 derivatives to investigate membrane lateral organization and membrane properties. And in some cases, we have attached fluorophores on the fatty acid of GB3s. And in some cases, the protein did not behave naturally. Normally, the protein is supposed to be located in the LO phase of the membrane, but when we put the fluorophore on the fatty acid, the protein was located in the LD phase. So in some cases, when you modify the fatty acid of GB3, you can, of course, unfortunately, alter the natural behavior of the protein, but that was not the case with the photo GB3. As you saw on the images, the protein was located in the LO domain following incubation with the supported lipid bilayers.
Okay, thank you. So there are a few more questions that have come in for Larissa, so I might just carry on with this for now and then hand over to Adam in a few more minutes. We do have plenty of time. So further grilling for Larissa. I think it's great that people are, have all these questions. So the next one is, how does photo GB3 remodeling of the protein-enriched membrane domains affect transported biomolecules, impacting drug discovery and treatment of degenerative diseases?
How does it impact? I did not understand the question.
Sorry, how does remodeling of protein-enriched membrane domains affect transport of biomolecules? And hence, how could it then affect sort of drug trafficking and any kind of drug treatments of diseases that are impacted by this too?
So in my case, I did not use the photogrammetry to design nanocarriers to transport biomolecules, but I know that some studies have been done with other types of photolipids, photophosphatidylcholine, but they use lipid nanoparticles that did not contain domains. However, when they functionalize the lipid nanoparticles with the photogrammetry, so the photolipid, they were able to achieve a control over membrane permeability, and upon trans to cis isomerization, they were able to release a cargo and to stop the release upon cis to trans isomerization. So to answer the question, yes, photolipids can be used to functionalize nanoparticles and for potential application in photopharmacology as carriers to deliver bioactive substances, but this is not something that I did. And to my knowledge, it hasn't been done with lipid particles that contain domain.
Okay, thanks. So I think there's a further question which I think is quite similar to that. So I might move on to the next one, which is just asking about whether you think the fluorophores included in these lipids could impact what we would consider to be normal lipid behavior or normal lipid trafficking if they were applied in cellular models. So I suppose, do you think the probes themselves might be influencing the observations you're making?
I think so. I'm currently doing other experiments to characterize the impact of the azobenzene on lipid-lipid and lipid-protein interactions. We are currently preparing a manuscript on that, and I can tell you that the azobenzene disturbs a bit the miscibility of the photogrammetry with LO and LD phase lipid, and it also modifies the binding affinity of the protein for the photogrammetry depending on the position where it is on the fatty acid. So yes, it can alter a bit the behavior, the interaction of lipid-lipids and lipid proteins. It can.
Okay, thanks. So the question is coming for Adam, if that's okay to switch over to you. So this says, how much of a role do lipid-protein interactions with trafficking proteins have on exosome biogenesis? For example, with ceramides or bis-monoacyl glycerols?
Yes, sorry, there was, my thing went funny for a second in the middle. How much of a, what was the question in essence? I missed the middle part of my...
Sorry, just let me, okay, so this says, how much of a role do lipid protein interactions with trafficking proteins have on exosome biogenesis?
A lot, I think. There was recent research with someone speaking about at the Extracellular Vesicle Conference, International Extracellular Conference, about GPI proteins being selectively enriched in some exosomes. In terms of trafficking proteins, I'm not quite certain what that category means, but lipid protein interactions are very, very important. We actually have at least two types of microdomains that are meant to form or meant to inform exosome biogenesis. There are ceramide ones, which I think were mentioned. You also get HRS protein scaffolds, which HRS will interact with clathrin and also with PI3P lipids, and that will stabilize the interaction, that lipid protein interaction. In addition, there's a whole family of proteins called tetraspanins, and any of those four-loop proteins actually play a big role in clustering lipids into kind of microdomains. and then also helping recruit proteins that wouldn't otherwise be directly inserted into exosome bilayers.
Sorry, could you just repeat the name of that family of proteins? I didn't hear it properly.
Tetraspanins. So 4 loops, tetraspanins.
Okay, thanks. I thought that's what you said. I've done a little bit of work on some tetraspanins, but just as a sort of technical assistant, so I don't know much about the biology of them. And I just wanted to check I hadn't jumped on something I thought I knew something about. That's really interesting. I think all of this stuff relating to protein and lipid interactions it's so cool to actually start to get to grips with it, because I think membrane protein research has really been so limited by ignoring lipids and pretending, like Larissa said, that it's just this little sea that the membrane proteins exist in, and actually starting to get into the detail of how they're all interacting and influencing each other is, I think, really important and probably quite vital for the field to actually move forwards. So yeah, it's really nice to see these tools. I'm just going to check in with the questions and see if any more have come in. And there's another question for Adam, if that's okay.
Yep.
So someone has said, thank you for the talks, to both of you, but to ask Adam about the RAB proteins and whether you've looked into their relationship to neurodegeneration.
Yes, so our lab is really split in half, with the much bigger half being focused on neurodegeneration and the secretory granules bit. Although they do seem like quite different topics, and I didn't cover it today, we found a very, very strange amount of interactions between the exosomes and the secretory granules. Yes, there will be a paper which we've just submitted to Cell, though that doesn't mean it's going to get in by any means. By lead author would be Dr. Preman Singh. I think it's on bioarchives, and that really goes into this. The highlight of that paper is that if you express human mutants, which you find in familial Alzheimer's disease, the cause of familial Alzheimer's disease, they disrupt secretory granule formation. They disrupt exosome formation, though that doesn't feature in full in the manuscript. And so yes, there's some really, really bizarre and fascinating links. If you express mutant tau proteins, you also get severely disrupted compartments and secretory granules in particular. Yeah.
Okay, thank you. So I think with that, I think we should probably wrap things up to stick to the timetable. Thank you again so much for those 2 talks. I'm sure that there are a few more questions which we don't quite have time to cover, so I don't know if we can send those on to you, and if you want to continue discussion with people later, that's great. I will leave that to Lucy and the Biochemistry Focus to organise. I've been asked to just say a few words to wrap everything up. So I think thank you also everyone for attending and submitting really good questions. There's nothing like actually being able to kind of get into the detail of things. I'm sorry if it felt a bit like a grilling. I hope it came across all right from the way I presented the questions to you. But it's, I think having these kind of discussions is really what makes science exciting. So hopefully it wasn't too stressful. Okay, so in fact, the notes from the Biochemistry say you can continue the conversation online by following Biochemistry and Portland Press Publishing on Twitter, or X, as we should now call it. If anyone has suggestions for future topics or speakers to feature in this webinar series, feel free to submit them. Please submit proposals via the Biochemistry. If you just go to the website, there should be the links to follow there. Also, upcoming webinars are listed on the website. I'm definitely feeling inspired to be an attendee myself on some more of these and make a bit more time for it in my schedule, and I hope other people feel the same after today's session. And there are also recordings of previously run webinars if people want to have a look at the topics and catch up on anything they've missed. Okay, so the next webinar will be on the 4th of July at 2 o'clock, called Improving Accessibility in the Lab, which will be, the invited speakers will be discussing approaches required to make research more accessible to all, which sounds like a great topic, and I'm probably something I should turn up for as well. And finally, I've been asked to say how important it is to stay connected and engage with fellow molecular bioscientists. I certainly feel like that my membership of the Biochemical Society dates back to my years as a PhD student, and I have to admit I first joined to get access to the travel funds, but of course it's grown into being much more than that as the years have gone on, and I've connected with people, and I'm now on one of the committees, which is a really especially for my career stage. I'm just finished being a postdoc, so it's been really great to feel like I'm making some steps forwards in my career by being involved with society and getting that feel of being one of the grown-ups occasionally. So if anyone listening is interested in participating in that, don't feel like being an early career researcher is a limitation. The Biochem Talk is really great at promoting us in our careers and helping us to find our way. So yeah, please engage with the society as much as you can. And of course, it's great for meeting fellow membrane people, fellow biochemists, and just finding a good network and friends in the area. So thank you very much to both of you for giving us your time and sharing your research with us. Thanks for all the attendees, and that's it. Have a nice afternoon, everyone. Goodbye.